Welcome to this video lecture series on red blood cells and white blood cells in medical biochemistry. This topic will be of 5 parts, with the first part focusing on the hemoglobin molecule. Before we move on to the main material, you should note that for exam purposes, you will need to read on these chapters from your Harper's textbook. I will also be basing my questions on my PowerPoint slides. I start off by defining the process of hematopoiesis, which, as you already know, is a production or manufacture of all the cellular components of blood and blood plasma, and this occurs within the so-called hematopoietic system, which includes the bone marrow, liver, and spleen. There are a variety of signals that trigger hematopoiesis. During the early stages, we have colony-stimulating factors, as well as the following interleukins that function as such. In the latter stages, particularly for red blood cells or erythrocytes, at the point where the myeloid progenitor cell is already committed to differentiation into the erythrocyte line, erythropoietin now becomes the primary hormone that stimulates their production. The receptor for erythropoietin is a dimeric receptor tyrosine kinase that does not have its own kinase activity, but instead borrows it from a cytoplasmic kinase called the Janus kinase or JAK. The phosphorylated Janus kinase or JAK subsequently phosphorylates another cytoplasmic protein called STAT, which dimerizes and enters the nucleus to act as a transcription factor that affects gene expression of growth factors necessary for erythropoiesis. Now, this entire signaling pathway is called the JAK-STAT pathway. By the way, in polycythemia vera, which is a rare disorder described by abnormal proliferation of RBCs that have high cellular mass, there's almost always a mutation in the JAK2 gene. Now, this mutation is one of the ways that the JAK-STAT pathway signaling can become dysregulated and cause the body to produce too many blood cells. Now, on the average, an erythrocyte has a lifespan of 120 days in circulation. Old or senescent RBCs are removed from circulation through phagocytosis that takes place mainly in the spleen. However, there are certain instances wherein RBCs are destroyed prematurely, and we call that occurrence hemolysis. And this happens because of underlying pathologies, which you will eventually learn about beginning in your second year. Now, if the degree of hemolysis occurs at a rate wherein the bone marrow is not able to keep up in terms of replacing the RBCs that were destroyed, then hemolytic anemia occurs. Now, hemolytic anemias can have extrinsic and intrinsic causes. When we say extrinsic, it means that the hemolytic anemia is caused by factors outside the red blood cell, such as antibodies from an autoimmune disorder, burns, certain medications, being transfused with the wrong ABO blood type, or an overactivity of the spleen called hypersplenism, which results in rapid and premature destruction of RBCs. Hemolytic anemias can also have intrinsic causes, and these are due to abnormalities in the function and or morphology of the component proteins of the RBC, such as hemoglobin, enzymes in the RBC, and structural or cell membrane proteins. Now, we are jumping ahead by first introducing to you the concepts of hemolysis when we haven't reviewed yet the normal characteristics of RBCs. Now, RBCs are devoid of internal organelles. Basically, in the development of an RBC, as it matures, it loses all organelles, including the nucleus. And because of that, it is unavailable to reproduce or undergo cell division in its mature form. For that matter, since RBCs are devoid of mitochondria, it generates its ATP exclusively via glycolysis. Now, a substantial amount of hemoglobin is contained in RBCs. And as we all know, the hemoglobin protein is the one responsible for binding oxygen as it gets transported in the circulation to areas of low oxygen tension, delivering maximum quantity of oxygen to tissues and aiding in removal of carbon dioxide. Now, this highly specialized function is thus what describes RBCs. Now, RBCs have a biconcave configuration, which is maintained by an extensive cytoskeletal network. Now, such configuration gives the RBCs a higher surface area to volume ratio, and it also allows them to fold over and squeeze through narrow capillaries. Now, having such configuration also aids in reducing the distance the gas molecules need to traverse or diffuse through to facilitate gas transfer. Heme is the non-protein part of the hemoglobin molecule and is the one that contains iron. The heme molecule is of the porphyrin class. Specifically, heme contains protoporphyrin 9, which is a cyclic prostatic group that consists of 4 molecules of pyrrole linked by alpha-methylene bridges. Iron is situated in the center of the protoporphyrin prostatic group. Its reduced form is ferrous iron, while the oxidized form is ferric iron. The planar network of conjugated double bonds is what gives heme its deep red color. And since protoporphyrin 9 is the most nonpolar porphyrin form, it is situated in the crevice in the hemoglobin molecule that is lined by nonpolar amino acid residues, except for 2 histidine residues in said crevice. Now, here we have the structure of protoporphyrin 9 as well as a graphic representation of the nonpolar crevice in which the heme molecule is situated in. As mentioned earlier, this nonpolar crevice contains 2 polar histidine residues, a proximal one and a distal one. Now, heme directly binds to the proximal histidine on one side. The other side of the heme molecule is where oxygen binds. The distal histidine residue interacts with this other side of heme, but is not bonded to it. Now, if we look closely into the interaction with histidine, we see that the central ferrous iron forms 6 coordination bonds, 4 with the nitrogens of each pyrrole ring, 1 with the proximal histidine, and 1 with oxygen. The globin polypeptide sequence of the hemoglobin molecule provides a microenvironment that allows for reversible oxygen binding, so that oxygen can be unloaded to tissues with low oxygen tension. Now, this microenvironment, while allowing reversible oxygen binding to heme, also protects the ferrous iron from getting oxidized to ferric iron, thus preventing the formation of methemoglobin. Now, this is particularly important because the ferric form of iron in methemoglobin is unable to bind oxygen. Methemoglobinemia is the medical condition that results from oxidation of the ferrous iron of heme to the ferric form, thus resulting in the inability to bind oxygen. Even though there is an abundance of oxygen dissolved in plasma, there is still hypoxia at the cellular level because the heme component of hemoglobin is unable to bind the oxygen molecule to be able to transport it. Now, methemoglobinemia can be inherited or acquired. In the inherited form, there is deficiency in either the quantity or activity of cytochrome B5 reductase, an enzyme dependent on NADH that is responsible for converting ferric iron to ferrous iron. Now, a variety of mutations in the alpha, beta, and gamma globin genes can also give rise to an abnormal form of hemoglobin called hemoglobin M. Most hemoglobin Ms involve a substitution of a tyrosine amino acid for either the proximal or the distal histidine amino acid found in the nonpolar crevice I mentioned earlier. Now, these amino acid substitutions cause heme iron to auto-oxidize, which results in methemoglobinemia. In the acquired form, on the other hand, nitrates, certain medications, and even reactive oxygen species, when abundant enough, result in the oxidation of ferrous iron to ferric iron. Let me now present to you the case of a 30-year-old female who was given lidocaine oral solution and benzocaine spray as topical anesthetic agents in preparation for an outpatient upper gastrointestinal procedure, probably an esophagogastroscopy. Within the next hour, she started having difficulty breathing, although her lung sounds are clear. On arterial blood gas analysis, her oxygen saturation was noted to be only 84%, even though her partial pressure of oxygen is well above normal. Even though the amount of dissolved oxygen in plasma is supranormal in amount, as reflected by her PaO2, the fraction of hemoglobin with oxygen bound to it is low at 84%. She was thus unable to transport oxygen to tissues requiring oxygen because of the low oxygen carrying capacity of methemoglobin, thus explaining her cyanotic appearance. Additionally, many patients with acquired methemoglobinemia would have arterial blood that is actually chocolate in color. Such patients are given a compound called methylene blue in order to correct this disorder. As you will note in the image on this slide, the right photo shows arterial blood from the same patient that is again red in color after the administration of methylene blue. As I mentioned earlier, cytochrome B5 reductase is an enzyme responsible for converting ferric iron to ferrous iron. I also mentioned that this is an NADH-dependent enzyme. In red blood cells, NADH is generated by the glycolytic pathway. Normally, cytochrome B5 reductase can reduce small amounts of methemoglobin that form randomly, probably due to reactive oxygen species that are generated during the normal process of metabolism. However, in acquired methemoglobinemia, this enzyme, along with other enzyme systems that normally reduce methemoglobin, get overwhelmed. Thus, methylene blue is needed to reduce ferric iron back to the ferrous form. This process oxidizes methylene blue in the process, which in turn can be dangerous. Thus, whenever one gives such a medication to treat methemoglobinemia, they should ensure that the patient is not deficient in glucose 6-phosphate dehydrogenase or G6PD. Known or suspected glucose 6-phosphate dehydrogenase deficiency is a relative contraindication to the use of methylene blue because G6PD is the key enzyme in the formation of NADPH through the pentose phosphate pathway, and G6PD deficient individuals generate insufficient NADPH to efficiently reduce oxidized methylene blue. I likewise mentioned that reactive oxygen species can also generate methemoglobin. As you'll eventually learn under the topic of reactive oxygen species, superoxide, designated as O2-, is one of the reactive oxygen species that get generated during the process of metabolism. Superoxide is formed when reduced flavins of FAD are reoxidized univalently by molecular oxygen. Recall from earlier topics that univalent oxidation or reduction of flavins leads to the formation of the so-called semiquinone radical. If they are not dealt with, they actually give rise to free radical chain reactions, thereby amplifying its destructive effects. The enzyme superoxide dismutase is responsible for removal of superoxide, and thus protects aerobic organisms against the potential deleterious effects of superoxide. The enzyme occurs in all major aerobic tissues in the mitochondria and the cytosol. Antioxidants such as alpha-tocopherol or vitamin E act as scavengers of free radicals and reduce the toxicity of oxygen. Now, superoxide dismutase converts superoxide to hydrogen peroxide. Hydrogen peroxide, however, is still toxic, so it must be converted into water and molecular oxygen by catalases in order to prevent the formation of hydroxyl radicals, which is another group of radical oxygen species. Now, this slide basically summarizes what I discussed in the previous slide. You can pause this video for a while to go through the information contained herewith. Now, this metabolic pathway here is what's called the pentose phosphate pathway. You will learn more about this when you get into the topic of carbohydrate metabolism. Now, the first enzyme in this pathway is glucose 6-phosphate dehydrogenase, which is an important enzyme because it catalyzes the reaction that likewise results in the generation of NADPH, which is necessary to restore glutathione in its reduced form with the help of the enzyme glutathione reductase. Reduced glutathione is found in high concentrations in cellular systems and plays a major role in detoxification of various free radicals and thus protects red blood cells from high oxidative stress that results in hemolysis. That is why patients with glucose 6-phosphate dehydrogenase deficiency, when they encounter strong oxidizing agents like certain antibiotics, for example, are unable to produce enough reduced glutathione, and very often this results in hemolytic anemia. G6PD deficiency comes about because of mutations in the G6PD gene, resulting in decreased activity of said enzyme. As I mentioned earlier, reduced activity of G6PD leads to decreased formation of NADPH and a decreased degree of reduced glutathione regeneration. Take note of the image beside the flowchart on this slide. Due to decreased levels of reduced glutathione, the oxidative stress results in the formation of Heinz bodies, which are actually composed of denatured hemoglobin. Their formation likewise alters the membrane structure of the affected RBCs, making them susceptible to ingestion by macrophages in the spleen, thus leading to hemolysis and hemolytic anemia. Now, there's actually another important enzyme that I have not yet mentioned, and that is glutathione peroxidase. I already talked about glutathione reductase earlier. Another thing about this enzyme is that it contains FAD as cofactor that transfers electrons or reducing equivalents. Recall that this enzyme functions by regenerating reduced glutathione by catalyzing the transfer of electrons from NADPH to the disulfide bond of the oxidized form of glutathione. Now, as for glutathione peroxidase, which is a selenium-containing enzyme, this cytosolic enzyme is the one that catalyzes the reduction of hydrogen peroxide to water and oxygen, as well as catalyzing the reduction of peroxide radicals to alcohols and oxygen, thus preventing the propagation of free radical chain reactions. I will end part 1 of this video series here. Please proceed to the next video, wherein I talk about the membrane structure of red blood cells.